So good morning. So hopefully people will still come. So we are in the disease control session. Progress in disease control, and my name is Helge SIEROTZKI. I'm one of the chairs, together with Anne-Sophie. I'm from Syngenta, working for 25 years now for the company in phytopathology as disease control research institute there. The session is in two parts. We have another part tomorrow about disease control, and the session deals with should shed light on the diversity and the strategies of disease control, but also for the challenges and the future perspectives. And by this, I welcome Anne-Sophie to introduce the first speaker.
Good morning, everyone. I'm Anne-Sophie Walker, coming from INRAE, near Paris. And I'm very pleased today to welcome Eric Chantelot, who is the director of the Technical Institute of Vine and Wine in France. And he will talk about his experience about implementing biocontrol in vineyards to manage powdery and downy mildew. So please, Eric.
Okay, thank you very much, Anne-Sophie. The French Vine Institute, AIV, is a national institute. We are not really a scientific laboratory, but much more an experimentation laboratory. That is to say that our work concerns the evaluation of solutions which should be directly used by growers. So we will try to develop some solutions, some new technologies to respond to the problems or to the evolution of the situation in the vine. So today we will present some information and global information regarding the vine disease and how we work to integrate new solutions, new biosolutions in control this type of strategies. How can we define the IPM approach in France? First of all, globally for all agriculture, since 2008, the French government decided to reduce the use of pesticides by 50%. That is to say that we are under constraints in order to develop IPM strategy to control pests, and not the IPM is not really an eradication of the pest, but try to control the pest. What are the different IPM components to go on that way? First of all, we can develop some approach on biodiversity development, grass cover, hedges, and so on. A large part of our work in the French Vine Institute is also by developing new resistant varieties. And that's a specific topic regarding this approach, but very promising. And then we can develop some biosolutions in the strategy. In terms of disease, today we will focus on the main two diseases in the vineyards, which are downy and powdery mildew. But we can also evaluate that we are focusing on increasing of the black rot pressure, mainly in relation with the reduction of the registered actives in the field. How can we define biocontrol in France? So there is all the biosolutions, which can focus on fungicide, but also biostimulant and so on. But biocontrol definition is very strict. It concerns natural substance which has really registered as fungicide. That is to say that the registration is similar to a conventional product for this type of biocontrol. And we can pretend in France as a biocontrol if there is absolutely no environmental or health effect of these active ingredients. That's the reason why, for example, copper because as an effect on earthrooms, cannot be considered as a biocontrol in France. A short list, not completely exhaustive, of different solutions. On fungicide, we can consider orange essential oil, for example, the potassium or sodium bicarbonate. We are also working on plant defense inducers like Cosoga, Cerevisiae, or Bacillus species. And we have a mixing of both, which are well known by a lot of growers, which are phosphonates, which are also fungicide agent and defense inducer. Now we will try to propose some evaluation of these different biocontrol, and then try to propose the way to integrate them in the strategy. On downy mildew, I take an example in 2020. On this graph, you have here all the treatment which has been done. The assessment, first assessment I will present. And we consider that at that time we had the first rain which causing contamination. So the first assessment is done just after this first contamination date. What we are comparing is an untreated here, and the product I presented previously on downy mildew in terms of low, in a condition of low disease pressure at that time, which is Cerevisiae, Cosoga, and essential oil. And as a reference, we compare with copper. On that picture, we can see that we can only identify one product which could have an effect on this low disease pressure, which is the essential oil. That is to say that it is a really fungicide approach and not the inducer. Cerevisiae and Cosoga does not show any real effect. If we consider another assessment later on in the season, after different rain causing contamination at the end of the season, we can consider that the effect is not really present, and we continue to compare that to make the evaluation that we can only consider essential oil in that test as a solution to integrate in the strategy. All the other products are not sufficient enough. If we are focusing now on powdery mildew, we also here there is different representation at the end of the season on bunches. You have been treated here with the 57% of severity attacks on bunches, and the efficacy of the different solutions we are testing. On that picture, we can also identify that only the solution who have a direct effect on the fungus, which is sodium hydrogenocarbonate or paraffinic oil, can be considered with a correct effect on powdery mildew on this test. The other solution, which is much more like defense inducer, has not sufficient effect, or nearly, for the first one, Cosoga, for example, no effect at all. So taking into account this different evaluation, this is just two examples I presented, but which is globally all the situation we can identify in the vineyards and the different tests we have done. What we can conclude on this is that biocontrol solutions are not sufficient enough. Biocontrol solution with direct effect on fungi seems to be more secure. That's the point we show regarding orange oil or bicarbonate on the powdery mildew. On downy mildew, we know that phosphonate is really the best candidate, and on powdery mildew, bicarbonate or paraffinic oil. So last part of my presentation, we'll try taking into account this first conclusion to propose some decision rules to develop a strategy based on the use on biosolution, biocontrol in the main approach. This represents the rules we define, and I will show you after that an evaluation of this approach in a test. On the low bottom, you have the disease risk, which is beginning very low to high risk disease. And depending on this approach, we propose different solutions. Only plant defense inducer when we are in very low risk situation. We complete this defense inducer with essential oil if there is some symptoms. And if we arrive with medium, high risk, we are complete the approach with phosphonate, which is the best biocontrol solution. And if there is really high risk, we can also complete our approach with copper to mix phosphonate with copper, for example, 300 grams of copper if there is medium risk, or 600 grams if there is high risk. Taking into account this decision rule, what we can observe, here you have on severity on leaves and severity on bunches, different area of evaluation, Alsace, Bordeaux, Cognac, etc. You have in green the strategy integrated biocontrol product, in blue the reference, and in red the untreated situation. The boxes here in color represent if there is in green similar effect as the reference, partially effect if it's in yellow, or no difference in comparison with the untreated, that is to say, no efficacy, for example, of this situation. So we can see in this picture during 3 years of evaluation that globally this type of decision rule is quite correct in order to control the disease pressure. Even if it's not perfect, even if there is some situation where there is no effect of our strategy and no effect and no difference, for example, in Bordeaux in that situation, no effect of the strategy compared to the untreated. But globally, we can consider that these decision rules are not so, could be interesting to develop them in the field. On powdery mildew, we decide also some specific decision rule based on the different stage of the development of the plant. First stage, before flowering period, we propose some solution based on the historic of the field and completed with modelization. So no treatment if previously on this field there is no powdery mildew, and go up to more important plant defense inducer plus desiccating product if there is a more important risk, and make sure we sulfur plant defense inducer and desiccating product if in the field historically there were a lot of powdery mildew. During the flowing period, we take no risk. In that decision rule, we systematically apply some sulfur. And then post-flowering, it will depend of, I show this, which names decitrate, which is a modelization, different type of product, always the same, taking into account the risk modelized by the model. Same representation than for downy mildew in this picture. And unfortunately, in 2 regions during 3 years, we do not have any powdery mildew in Languedoc and Val d'Oise. It's difficult to make an evaluation. But on the other approach, we have globally the same response and evaluation. That is to say that in some situation, we have an efficiency quite similar to the reference. But on some situation, next in that on leaves, on Bordeaux, the situation, the evaluation of the strategy is not different than the untreated strategy. So all this approach in terms of conclusion, this integration of biocontrol is mandatory in a program to develop IPM strategy and to manage, to reduce the use of pesticide for 50% as the government decided more than 10 years ago. Biosolution, biocontrol are not sufficient on their own, so we have to combine and to define decision rules to combine these biosolutions with other active to maintain the efficacy and the security of the program. But the problem of this approach, of this decision rule, is that this combination of different biocontrol is not acceptable in terms of cost. So it's very difficult to convince growers to develop this type of strategy because of an economic approach. Growers take more risk with 3 times the cost of a normal application. So for the future, we will work, we continue to develop some experimentation on biocontrol, trying to better understand the mode of action of biocontrol products, to identify new solutions, and we have a lot of discussion and partnership with some society which develop companies, which develop some solution. And we will try to work also on new mode of application. And the fact is our strategy for the time being is based on conventional approach. That is to say that we applied each 7 days, each 10 days products as it was done by the past. And we identified that probably for plant defense inducer, for example, it could be interesting, it could be to try to identify if there is better efficacy if we are working by reducing and apply 3 times a week or 3 times very short in the beginning of the season. So we are also working on this new mode of application in order to increase the efficacy of this solution. Thank you very much.
Thank you, Eric, for this nice talk. So there are many questions in the chat. Maybe I will group the ones concerning the applications. So, how many times were the vines sprayed with oil and all the biosolutions? And also, what were the modes of application? For example, how do you apply for the essential oils or copper? Does it demand a high quantity for a field? A high quantity for the field when you apply such a compound? Because the dose rate were not always mentioned.
Yeah. The application of product is done with spraying system, pneumatic systems with low volume of application on this test. The dose rate we applied is the dose rate recommended registered, in fact. So if it's 2 liters, we apply 2 liters of product in mainly 100 and 150 liters per hectares. That's the point. We want also to work on the volume of application because we have some feedback from other countries which are working with other volume of application, much more 400 liters, 600 liters. And it seems with this type of volume of application, sometimes there is some actives, biocontrol actives, which seems to be more effective with this volume of water. So we will want to try to test this. But the problem at the end will be to develop the solution in the field because growers try to reduce the water volume in order to reduce the time to fill up the tank. So it will be difficult if we manage to find some solution saying you have to work with 500 liters, that it will take 5 times the time they're taking for now to apply it. So it's not very easy to develop this solution.
And how many sprays in your trials?
The sprays for downy mildew on the picture was around 10. It was all the point I mentioned.
Oh, okay.
And for powdery mildew, we are about 3 before flowering period, 3 during flowering period with sulfur in our strategy, and 2 or 3 after, so more or less 8 to 9 treatments.
So, you want to go on?
How do you define the risk class for downy mildew?
The risk is defined with a different type of model. So we use at our institute one type of model which is developed in the field for growers also, but we can use other type of model which define the risk. For downy mildew, it's quite simple because in fact during the season, after the first contamination, we know that each rain can occur some contamination. So it's quite easy to define, and we try in all our trials to apply product and biocontrol just before the new rain which will occur contamination.
And so what was the pressure in your trials? Over the 3 years, was the pressure constant?
On powdery mildew?
I guess on downy mildew. In the question, it's about downy mildew.
What I mentioned on downy mildew with first slide I presented was a slide concerning one test, and the pressure was over nearly 100% in the untreated.
And there are also 2 questions about the type of compounds you have chosen. So there's one about phosphite, which is a synthetic compound, then a major metabolite of aluminium phosphatyl. What is the advantage of using phosphites in relation to other fungicides in terms of toxicity to humans? Is it a real biosolution?
Good question. In fact, for sure, phosphite, globally, is a synthetic compound, but which has been included in the list of biocontrol. So it's a good situation for growers because it's quite efficient, but I completely agree that it can be really considered as a biocontrol, biosolution, as well as laminarine or Kosoga I presented. So it's a bit tricky situation because it is registered as a biocontrol, but much more as a conventional because it was used as phosphatyl, as it is mentioned in the question for a long time ago. So we know it's not really a biocontrol.
And
are there any questions from the audience here? Just to open the floor. Otherwise,
if not, there is one last in the chat about the use of microbial antagonists, which you haven't mentioned. So there are Ampelomyces, Bacillus, Pythium. Do you also evaluate these solutions?
We have made a lot of evaluation of other solutions, but I mentioned in this presentation only the solutions which are already registered.
Okay. The other ones are not registered. Okay. Well, thanks again, Eric.
Thank you very much.
thank you very much for this interesting talk about how it is in reality. And now welcome Oscar Villanueva from the Agriculture and Agri-Food Canada Research Institute and optimizing the field-based control of Phytophthora capsici. I can click for you here.
Hello everyone. Hello everyone, and thank you for the introduction. My name is Oscar. I am a PhD candidate from Brock University, and I also work in Agri-Food Canada. So today I'll be talking about my field experiment and how can we use the multiplex ddPCR to quantify biological and pathogen populations. So to begin with, I would like to start with introducing my pathogen, which is Phytophthora capsici. It's an omicide that occurs worldwide. It affects many vegetable crops and creating serious crop losses. As we can see here in the map, it's all over the world. And Phytophthora capsici is a causal agent of Phytophthora disease. It affects many crops, such as pepper, tomato, pumpkin, squash, and cucumber. When ideal conditions are present, which are moist soils, temperatures between 24 and 29 Celsius degrees, it develops the following symptoms: pre and post-emergence damping off, wilting and fruit rot, as well as the root rot. In this picture, I don't know if it's kind of clear, at the back here we can see that there's a fully rotten plant. We have something wilting here. This is a pepper plant, and in the back over here, we have like a healthy plant. And all of these three plants are on the same bed. So meaning that we can see how the pathogen travels around. Since it's an omicide, it creates zoospores that are able to travel with water. So in Canada, this pathogen was first discovered or reported in Essex County in 1994 in peppers and tomato in fields. Then it moved to Quebec in 2001, and then it was recently reported in British Columbia in 2007. When proper commercial management is used, more than $100 million are used to control these creating losses annually. But the improper commercial management creates the complete loss of this crop. So over the year, different types of controls have been used, such as the chemical control being the use of fungicides. We have the cultural control, which is the use of raised beds, plastic covers, monitoring the irrigation frequency. We also have biologicals control, which are the product serenade that has the active ingredient of Bacillus subtilis. And recently in Canada, we have this new product, Fortrix, which has two active ingredients. There are two trichodermas. One of them is Trichoderma gamsii and Trichoderma spirillum. Something interesting about these two active ingredients, Trichoderma gamsii works only on its ideal condition. It's 15 Celsius degrees, and Asperillum is 25. This would be relevant in my results that I'll be showing you. But over the years, when people have used these types of control individually, they haven't been able to control it. So that's why my project or research is about has the main two objectives. With one of them, the first one is to control Phytophthora disease in pepper by developing an IPM practice, and also to use multiplex ddPCR to follow the population dynamics of Trichoderma gamsii, Trichoderma spirillum, and Phytophthora capsici. So, and this project is being divided in two main components. My field experiment, where I combine different cultural practices, such as the raised beds, the irrigation frequency, plastic covers, with chemical controls, such as the fungicide forum, with the active ingredient of dimethamorph, and also biologicals, which are the Fortrix, the new product in Canada with the active ingredients of the trichodermas, and serenade. Then these results will be analyzed using a new technology, digital droplet PCR, and I'll be multiplexing and looking for these three main microorganisms, which are the two trichodermas on Fortrix, and as well as a pathogen, Phytophthora capsici. To start with, I want to show you my field experiment. So once again, I have two types of water irrigation. I have 1 inch per week or 2 inch. I have two types of beds. These were raised beds with plastic covers, and then flat beds, and then the treatments were a positive control and negative control. Fortrix, the combination of Fortrix and the other biological, and Fortrix and the fungicide, the chemical fungicide. And here are some of my results. In these results, I am showing you 2 years of results. And on the x-axis, we have the different treatments and the positive control. And on the y-axis, we have the area under the progress disease curve. As we can see on the first year, all the treatments work. Fortrix on its own and in combination of the other biological, they work reducing the Phytophthora disease. But in 2022, only the combination of Fortrix with a chemical, the fungicide, were able to reduce this, reduce the Phytophthora disease. As we can see here, we can say that on 2021, it worked perfectly. But something that I can say it is like on 2022, we had a bunch of rain that it didn't allow us to really see the effects of this product. Then here on the next results, I am showing you the effects of the types of bed with different irrigations. And once again, we can see that on the flat beds, there was less disease. And this could be because the plastic covers do not allow the water to go out of the bed, making ideal conditions for Phytophthora to reproduce and grow more. So that even though we have these results, we haven't looked at how many pathogens we have in this treatments. So that's why I'm doing a multiplexing on ddPCR to know to quantify these populations. So the way multiplex ddPCR works is that each target would get a different label. In this case, Phytophthora got the hex probe. And for the two trichodermas, I am multiplexing them on the same channel using a different concentration of the probe. As we can see here, Trichoderma gamsii has a FAM and a FAM low, and Aspergillum has a FAM high. What the machine does is identifies each target based on their fluorescence, allowing them to be in a different quadrant. As we can see here in the picture, this is where Aspergillum would be put every single time we get a read, gamsii, and as well as capsici. So here's a workflow on how the ddPCR works. I have taken samples of root and soil from the rhizosphere, goes through the droplet generation, creating more than 20,000 droplets. Each droplet represents a single PCR reaction. After the PCR amplification, the optical reader will take each droplet, read it, and assign them into the different quadrant with a different target, and then plotting those results into a graph and giving the quantification. Here I have some of the preliminary results just to show you how a reading looks and how can we quantify these microorganisms. In these graphs, I have two results. The first one has a 90% plant infection, and this is a plot that comes from the ddPCR, having the FAM on the y-axis and the hex on the x-axis. We can see a high amount of Phytophthora capsici interacting with a very low amount of Trichoderma gamsii and Aspergillum. And on the other graph, we can see an 18% of plant infection, and it correlates with a lower amount of Phytophthora capsici, and again, a higher amount of Trichoderma aspergillum. But if we can see here in the treatments, this is only the first one with a 90% is Fortrix on its own, and here we have the combination of Fortrix and Serenade. These results are preliminary. I still have to take in consideration different factors because this is a field experiment, so there's the interaction of weather, temperature, and once I get that, I'll be able to really make a correlation with these results. So I just want to finish my talk with some conclusions. We can say that Fortrix worked on 2021 in reducing Phytophthora disease, and that by using ddPCR multiplexing, it would allow us to know the quantification of the three microorganisms that I'm using, the two trichodermas and the Phytophthora capsici, in one single sample. And yeah, I want to highlight the usefulness of doing multiplexing ddPCR, which is a technology that is very unique and very useful in these types of experiments. And thank you for your time.
Thank you very much, Oscar, for this presentation. Are there any questions from the floor? Yes, there is one.
Hi.
Hello.
Yeah, I have a question about how you interpret that droplet PCR results, because so you're detecting DNA. So for your pathogen, yeah, I don't see issues because it's coming in naturally. But your trichoderma, you're putting in. So basically what you're detecting is DNA. So it's your input. However, you not know if that trichoderma is actually alive or interacting or anything. So is there a way you could normalize your data in a way that you could make this relative to actually it is alive and active? Because indeed, if I put trichoderma in a soil, yeah, with PCR I will detect it. However, if I do some plating, I will see that there is changes in populations. So like, yeah, I'm a bit skeptical about the PCR approach.
Yes, I understand your question. So It's kind of interesting because what we have been doing is making a standard with the amount of product that we put in there and what reading are we going to get in the ddPCR. But we haven't looked at if it's alive or not, so maybe that could be the next stage for our experiment. Sorry about that. So thank you for that question.
Yeah, thank you for the nice talk.
Thank you.
If there is no other from the audience, there is one you have tested for red drugs. Haven't you done the same for serenade and on the bacillus as well, or is this in the plan?
Yes, right now because we're on the optimizing stage, we only did the 3 main products that we're using, or the 3 main microorganisms. The next stage is to add an extra label to maybe look for Bacillus subtilis into this mix. Yep.
There are many questions about the technology, about the primer, and but what kind of material did you use? The genomic DNA or from the?
Yes, we do genomic DNA. We do an extraction from the roots, and then we do an extraction from soil because we want to know what is stuck in the roots, and we want to know what is around in the rhizosphere. So from there it goes into the, we mix the primers and the labels, which are the probes. One of them Hex, and 2 of them the FAMs, and with different concentrations.
And perhaps one final one, and then we close. How sensitive is your assay? Have you also tried asymptomatic plants, which you have?
Yes, we have
disease, but not yet visible.
Yes, because we are working on the range of the detection of the ddPCR. We have to test asymptomatic to see if even the very least amount of Phytophthora will start giving us a signal. So yes, that has been tested, but because I haven't finished my analysis, that we're not put into this presentation. Yeah.
Good. Thank you very much again for the talk. So then the next talk is given by Kari PETER. Yes, she's coming from the Pennsylvania State University, and she will report on fungicide sensitivity in Colletotrichum species.
Okay, all right. So thank you very much to the committee for the invitation to speak to you today. The work I'll be talking about is from my grad student, Philip Martin, and he worked on bitter rot for his PhD, and this fungicide sensitivity story is just one of many stories. And this is already published, and it's in open access, so FYI. So in 2018, we had the worst bitter rot epidemic to hit the mid-Atlantic, and as a result, everyone was asking questions, especially the growers and us as researchers. So the biggest questions were there fungicide resistance occurring among the Colletotrichum isolates out there? And also, what was the relative sensitivity to the fungicides in general that we were using in the field? And so for over 2 years, we collected over 500 isolates from 38 orchards, and this was about 5 states, with majority in Pennsylvania. And we identified 8 different Colletotrichum species within the mid-Atlantic, and they fell within 2 different species complexes, the Colletotrichum acutatum species complex and the Colletotrichum gloeosporioides species complex, with the most dominant species being Fiorinae and Chrysophyllum. And so Fiorinae is acutatum, Chrysophyllum was in gloeosporioides. And we also were collaborating with Dr. Sergeant Achimovich at the Cornell Hudson Valley Lab. He was there at the time, and they were seeing similar trends as well with regards to the species complexes. So of those over 500, we took 220, and we then screened them for fungicide resistance, and that's the breakdown of the species, the number of species, of isolates per species that we did. And so we tested against 11 different active ingredients, which covered 6 different modes of action. And so we chose these 11 different active ingredients based on the specimen label that had bitter rot as one of the diseases it controlled. And so these fungicides exist, we use the formulation, they exist either as a standalone or in a premix. And so we determine the screening dose, and now I'm quickly going to go through the histograms of the relative mycelial growth for each. So first, FRAC group 1. The take-home message is that the sensitivity varies by fungal species, with the Gloeosporioides complex being more sensitive to FRAC 1, whereas the Acutatum complex isn't as sensitive. We did identify some resistant isolates, but it was just in one species, and that was the Colletotrichum siemensii, and we did confirm the beta-tubulin mutation. FRAC group 7, take-home messages are 1, the species, they behave similarly between the active ingredients, but there was variation among the active ingredients, with the benzovitaflupyr being being the strongest as far as suppressing growth. For FRAC group 9, this was mostly insensitive, but the species behave similarly. Even in our dose response curves at 1,000 parts per million, it really wasn't doing much. As far as FRAC group 11 goes, so there's some interesting take-home messages here. 1, the species were behaving similarly. 2, we saw differences among the active ingredients. And 3, we did identify resistant isolates. In the case, again, it was Colletotrichum siemensii, again, and it was just 2 isolates that we identified. And there was partial resistance, so it was only resistant to pyraclostrobin and trifloxystrobin. It wasn't resistant to croxamethyl, and we aren't quite sure why that was, and we didn't delve into it any deeper beyond that. But we confirmed it. We confirmed the resistant isolates with the mutation. FRAC group 12 and 29, they both did very well. FRAC group 12, the species complex varied a little bit, but I will resurface, I'll come back to this. Fluazinam, the FRAC group 29, did a very good job at controlling. So then we delved in a little bit deeper where we wanted to look at the EC25 and EC50 for all the modes of actions that are labeled for apple in the Mid-Atlantic. And so in addition to the first 11, we took an additional, another additional 11 for a total of 22. And so in this case here, we have all 22 fungicides, EC50s on top, EC25s on the bottom, and the parts per million's pretty large. So take-home messages here are 1, the species are responding similarly between the FRAC groups. We see diversity among the FRAC groups, and we also see diversity within the FRAC groups. So Philip drilled down even further and took the top 14 best performing fungicides, and that's where the red arrows are. So looking at a little bit deeper, so the EC50 values and EC25 values, they're a little bit smaller. And so what we're seeing here is that in the case of FRAC group 3, we're really seeing a lot of differences. Difenoconazole and mefentrifluconazole are looking to be the best fungicides. We're seeing differences between benzovitaflupyr and penthiopyrad for FRAC 7, and definitely differences between the FRAC 11s, particularly pyraclostrobin and trifloxystrobin. But by trifloxystrobin, we're seeing really big differences between EC50 and 25. And fludeoxonil and fluazinam 12 and 29 are doing pretty well. So we then took this a bit further into the field. Okay, so we have this concurrently, we were doing this EC25 and EC50 data. How does this translate to what we see in the field? So we did 2 field evaluations at Penn State and Pennsylvania, and also in New York, in Highland, New York, and the Hudson Valley, and we tested different fungicides and different FRAC groups. And we also added a couple of organic options too. That's FRAC group 19 and P3. And so first at Penn State, we looked at 2 different cultivars, Honeycrisp and Cameo. As you can see, there's drastic differences. We looked at harvest and post-harvest for Honeycrisp. It ranged from 0% to 100% incidence. For Cameo, it was much less. The max was about 10%. And so what we're observing here is that the different fungicides, we're probably seeing other factors influencing efficacy. We do see differences with the FRAC 7, the penthiopyrad and benzovitaflupyr, where the penthiopyrad didn't do as well in the more susceptible variety of Honeycrisp. Pyraclostrobin did well, fluazinam did very well in the, both cultivars, especially in Honeycrisp, and Captan's always our go-to, and we always include that. So as far as take-home message here is that fungicides the efficacy might be influenced by definitely cultivar susceptibility and disease pressure. We saw similar trends in Highland, New York, whereas they also looked at 3 different varieties, Royal Court, Cameo, and Honeycrisp. They had 2 different harvest dates, an early harvest date and a later harvest date. The most interesting data is at the later harvest date for Honeycrisp. And so they were seeing similar trends, such that the FRAC 7s in general, not that great. However, when we see penthiopyrad down here and benzovitaflup here, we see it performing similarly to how we saw it at Penn State, where benzovitaflup here was doing a much better job. Then we have the FRAC group 3, which isn't labeled for bitter rot. It looked good in our EC15.25, but the mefentrifluconazole, didn't really come up to snuff as far as dealing possibly with those latent infections. One thing of to note, interestingly, trifloxystrobin. Trifloxystrobin in our EC50, particularly EC50 data, says trifloxystrobin is not going to work, but we're seeing it work really well in the field. And anecdotally, I will say I do have data that trifloxystrobin does work in Pennsylvania. So as far as EC25, this is a paradigm shift. EC25 is a better predictor of field efficacy to control bitter rot. So I'm breaking down 3 different FRAC groups here to illustrate this. FRAC group 7. So we have benzovitaflup here on the bottom line, penthiopyrad at the top line. When you look at the 25% growth, this is where you see the difference. And at 50% growth reduction, it's a lot more closer. But this really reflects to what we see in the field. FRAC group 11, we see pyraclostrobin on the bottom, trifloxystrobin in the middle, and then crisoxamethyl at the top. So trifloxystrobin and pyraclostrobin at reducing 25% growth, it's performing similarly. But at 50% growth, this is where you see the discrepancies. But at that 25% growth reduction, that's what we're seeing out in the field. FRAC group 12, back when I talked about the resistance data, we saw differences in the Gloeosporioides and Acutatum complex. But when you look at the 25% growth reduction, they're controlled similarly. So in the field, I expect that's how it's going to be. So as far as the relevance of EC25, we need to be thinking about the disease triangle. So we always think of fungicides as killing that pathogen, but actually we need to be thinking about fungicides impacting the right conditions. So Colletotrichum needs surface wetness hours to germinate and penetrate the fruit, and the dependent variable there is the ideal temperature. So what the fungicides are doing, as far as the ones that as far as encourage the suboptimal growth, is that it's destroying those right conditions by creating basically the illusion of lack of suboptimal growth. And so what we're seeing here is that by slowing down growth and acting as this suboptimal temperature, we are extending the number of wetness hours required for infection, hopefully past the hours the fruit is actually wet. So in this case, EC25 is a better predictor under normal conditions because it's a fungicide concentration that's going to suppress a growth. EC50 is that may be a better predictor for more severe disease conditions where you have greater wetness hours and ideal temperatures. So to quickly go through the conclusions here, increased incidence not due to fungicide resistance. We saw a very low frequency. It was only in 1 species. We saw it in FRAC 1 and FRAC 11. The dominant species were ineffective. As far as our in vitro testing of every single mode of action that's currently available, we saw small variation of sensitivity between the species, wide variation in efficacy of the fungicides within the same FRAC group. And in general, to majority of the fungicides, Colletotrichum isn't sensitive. So that really restricts the number of fungicides we can use to control the disease. We have this paradigm shift as far as looking at EC25 values as better predictors of field performance under normal conditions. And as far as the best fungicides, I have them listed there. The FRAC 3s, I have question marks. I'm still dubious about this. I don't recommend them to growers, but I think we need to look at them a little more carefully. There has been some research of possibly combining the two, that maybe 2 of them is better than 1. So with that, I've got a lot of folks to thank, especially Philip for all of his hard work, the funding agencies, the apple growers, and with that, I'm out of time. So I thank you for your attention.
Thank you very much for this high-density data presentation. Is there any question from the audience? If not, there are some on the chat. Did you have the relation to the baseline efficacy? So that because often there's a confusion on resistance and real efficacy in the field.
Yes, that is all in the paper and in the supplemental data, and I just didn't have time to show that. But yeah, I did confirm. Yeah, we did confirm that. That's in the paper. As far as the relationship of that, yes.
And the other one is about the interesting difference between the Colletotrichum species, that some acquire the G143A and others not. Have you checked the intron in the cytochrome B present?
So we have not. So we haven't gone deeply because we only have those two isolates as far as for the FRAC11 resistance. So we got as far as saying yes, it's there, but we haven't gone beyond that at the moment.
Yeah. And have you information about historic sensitivity from very old Colletotrichum species or isolates?
Oh yes, I forgot to mention that when we were testing our fungicide isolates, I forgot to mention we also included isolates that had not been exposed to fungicides, so which came from organic orchards, and also from the forest. Particularly, we found Colletotrichum fiorinea, which had never in the forest. It's a leaf endophyte, so those ones, and also we had reference isolates from various folks that donated them to us. So we had like 30 extra isolates in addition to the ones. And they all were susceptible. Like the organic ones or the ones that had never seen fungicides before, everything was still susceptible.
And then there are one question about the EC50 paradigm shift. Does this also hold for other Colletotrichum diseases in fruits?
That's a good question. That would have to be validated. But I would say yes. I mean, particularly as far as under normal conditions. In this day and age, normal might be changing. But it would be worth validating that. But I would suspect, yes, that EC25 could be extrapolated to other systems.
And then there is one from the floor. Thank you.
Yes, thank you very much for a very nice presentation. My question is relating the strobilurins. I'm understanding that the EC50 was measured fungicide sensitivity by mycelial growth on agar. My recollection is that with strobilurins, the effect may be more on spore germination rather than hyphal growth, so they may appear insensitive in culture, but that might explain better efficacy in the field potentially. Is that something you have looked at, spore germination versus hyphal growth?
Yeah, we did not attempt the spore germination due to getting spores to do that evaluation, but we did discuss that as far as particularly with the strobilurin class, but we did not explore that. But that's a good point to make. And I believe that he did address that in his discussion of the paper. Because I've reread the paper at least half a dozen times before this meeting, so to make sure I remembered everything he mentioned. So.
Oh, thank you.
Okay, when there is no other question, thank you very much, Kari, for this very comprehensive.
And now we're really pleased to welcome Dolores FERNANDEZ-ORTUNO, Lola, who's professor at the University of Malaga in Spain, and she will detail RNA interfering with pathogen virulence to manage grey mold and cucurbit podry mildew.
So thank you very much. So first, I would like to thank the organizing committee of this section for inviting me and giving me the opportunity to present part of the research work that we are doing in our lab in Spain. So in our group, we are studying two fungal diseases. In one hand, the grey mold disease produced by Botrytis cinerea, that is a necrotrophic fungus. It's very important pathogen worldwide because it's able to attack hundreds of hosts at harvest and post-harvest, causing important economic losses. On the other hand, the cucurbit powdery mildew produced by Podosphaera xanthi, that is a biotrophic fungus, able to develop this structure of parasitins called Austorium, and with this structure is able to take nutrient from the host and release effectors to modulate plant defense mechanisms, being as well a serious limiting factor for cucurbit production, not only in Spain, also in the rest of the world. Our growers trying to control both diseases using different strategies, but the main one is the chemical control. For this reason, in our group, one of our research lines is to know more about the fungicide resistance situation that we have with the main classes of fungicide that were or are registered in our country. For that, we are doing biological assays, fungicide sensitivity assays, and molecular assays. And depending on the results that we obtain, we provide recommendation to our growers. The problem is that the majority of the studies that we are carrying out, always we observe high level of resistance to the main classes of fungicides, especially DMIs, also QOIs, MVCs, and the recently introduced SDHIs. This is not surprising because both pathogens, according with the Fungicide Resistant Action Committee, are high-risk pathogen for fungicide-resistant development. In addition to that, we have another problem, and it's the reduction of the diversity of fungicides that we are going to have in few years. All of this according with the European Union and the European Green Deal to the Farm to Fork strategy. For this reason, the other research line that we are carrying out in our lab in Spain is to develop sustainable alternative of control. For that, we are focusing on oligonucleotides in forms of aptamers and dsRNAs. We'll try as well to protect these oligonucleotides with the use of nanoparticles, with the idea to be used in the field and control in a short future both diseases. Focusing in the first one, in the use of RNAi-based fungicides, this is a potential tool for fungal disease control. In our lab, we implemented through the use of the spray-induced gene silencing. That means that we are able to develop these RNA solutions and spray onto the lips of our host. And these dsRNAs are designed to silence essential genes for fungal development or pathogenicity. So thanks to the cross-kingdom RNAi, we are going to be able to silence the mRNA that were the dsRNA designed at that moment. So I recommend you to read this interesting review about the RNAi strategy that we recently published in our group. In Botrytis cinerea, we use at the first moment this strategy using as potential target the Botrytis cinerea SOT1 gene, that is the superoxidol dismutase 1 involved in ROS machinery mechanisms. And we know, as previous study for other authors, that this that has a key role in the pathogenicity of Botrytis cinerea. As you can see in that publication for other authors, the mutant in this target gene is not able to produce the same necrotic area of the wild type. So, as part of the PhD thesis of Alba Lopez, she carried out a detached leaf assay. For that, she made first the dsRNA constructions and then applied onto the tomato leaves, detached tomato leaves, and then put in contact with a drop of Botrytis cinerea conidia suspension. And then we tested several parameters. So as you can see in that picture, 5 days post-inoculation, we use as control water at fungicide, the registered fungicide fludeoxonil, and then we apply the dsRNA at 100 and 300 nanogram per microliter. And as you can see, we obtain a disease reduction around 70%, more or less, in both concentrations. Also, we measure by qPCR the fungal biomass, and also was reduced, especially at 90%, at the concentration of 100 nanogram per microliter. Also, we measure the conidia germination in vitro assays. For that, she put together the DNA RNA solution with the conidia suspension and then observe under the optical microscope. And as you can see in the pictures and in the graphic, the conidia germination of Botrytis cinerea was reduced by 52%. But also, we wanted to know if the fungal cells were able to take up the dsRNA. And as you can see in these pictures, it was possible, and the dsRNA is able to enter inside the cell. With the cucurbit powdery mildew, we developed the similar strategy. But in this case, as we have a lot of information in our group about the genome and the transcriptome of Podophera santi, in the study carried out during the PhD thesis of Laura, we identified and select essential conserved and non-annotated proteins in Podophera santi that presumably were involved in essential functions as, for example, efflux transporters, glycosylation, or respiration. So in that publication, at the beginning, we tested via 6 only 3 of targets, 3 of them, of the 6. So we designed the dsRNAs, then apply in a battery of concentration, and then 24 hours later, we apply the Podophera santi conidia suspension. Then 12 days post-inoculation, we observe the results, the development of the Podophera santi onto the melon leaves, and as you can see here, the results were very good, with our disease reduction greater than 80%, especially at the concentration of 10, 20, and 30 microgram per milliliter. We use in that case as positive control the Podophera santi beta-tubulin gene, because we know that is an essential gene for fungal development as well. And a negative control, the water. Recently, this year, during a final degree project, we also tested the other 3 targets. So we developed a similar assay, and as you can see, also we obtained a disease reduction. In this case, was not as higher of the other publication, but still pretty good results, with around 65% of disease reduction, especially for this efflux transporter gene. But also, we wanted to know, or we wanted to target some effectors related with the chitin. So as you know, the chitin is an essential component of the fungal cell. When chitin is recognized by the plant with the plant chitinase, oligomers of 6 subunits of Helge are released. In this case, these chitin oligomers are recognized by the plant receptors, and when it's recognized, the chitin trigger immunity is active, so the plant defense machinery is active. What happened? That in previous studies carried out in our group, we were able to see that Phytophthora santi can manipulate this immune response by several strategies. For example, with the use of the UCAS effector with chitinase activity is able to cut the chitin oligomers in subunits of less than 6. So in this case, it's not able, the plant, to recognize these oligomers with the receptors. Also, with the chitin, the acetylase is able to convert chitin to chitosan. So the chitosan is not recognized by the plant receptors. So during the PhD of Nisrin, we are studying these target genes, and in this case, we carry out a similar experiment that I explained before. We use, again, as a control, the beta-tubulin gene and the target genes that I mentioned before, alone or in combination. And as you can see in the graphic and in the picture, the results were around 50%, but especially the disease reduction was higher when we apply both dsRNAs together. On the other hand, as I said before, we would like to protect these dsRNAs in nanoparticles, because we would like to use in field conditions, and we would like to improve their efficacy and delivery in natural conditions. So for that, at the moment, there are several nanoparticles published in the literature, but at the moment, we are using the clay particles following the nice and very interesting publication and recommendation of Nina Mitter, and also the carbon dots in collaboration with Leonardo Velasco in the IFAPA in Malaga. We only have, at the moment, preliminary results, so we apply naked or in combination with the clay particles, the dsRNAs, in this case, for one of the CNAP target of the Laura publication. And as you can see here, at the moment, we only obtain more or less the same results when we apply the same disease reduction when we apply the dsRNA naked or encapsulated 15 days post-inoculation. So the idea is to leave more time the experiment to see the efficacy. We did the same with the carbon dots, and in this case, we obtain better results. So as you can see here, we observe higher reduction of the disease when we apply the dsRNA together with the carbon dots. So as you can see in this graphic, when we apply the dsRNA naked, they lost efficacy after from 10 days until 20 days, a little bit. But then in 20 days, still were very effective when were protected in the carbon dots nanoparticles. Then we have as well, as I mentioned before, the use of other oligonucleotides that are the aptamers. So the aptamers are RNA or single-strand DNA that are able to fall into a unique 3D structure able to bind specifically the target protein. They are like chemical antibodies very, very used in the clinical sector. So we would like to use in the agricultural sector for the first time. So I don't have time to talk about that, but I recommend that you know now, because the poster was posted on Monday and Tuesday, but we were the winner of the poster prize, so you are going to be able to see this work that is from our PhD student Alba, and it's very interesting because in here we compare results using 6 aptamers targeting the SOT1 protein. So in summary, I don't have any doubt that in a short future, I hope so, that we can introduce the use of RNA-based fungicide to control both diseases in the field. But at the moment, it's going to be necessary at least to produce these molecules with lower cost, because at the moment it's very expensive if we want to perform field experiments. So with that, I would like to thank our group, especially my teammate, Alejandro Pérez García, our lab technician, Yandira and Virginia, and our PhD students, Laura, Nisrin, Alba, and Isa, because working with them or supervising them is a really nice work for me. And our funding support, the Junta de Andalucía, European Union, and the Spanish government. So thank you very much for your attention.
Thank you, Lola, for this very nice talk. There are plenty of questions in the chat, so I try to group them. There are some questions about the stability of the RNA. So what do you do to stabilize them, to avoid degradation? What is the shelf life?
We really don't do anything too much, because when we apply, we use a nuclease water solution, and that's it. At the moment, in the experiment that we were carrying out in chamber condition in plants, 10 days, 20 days after inoculation, they are still working. So we are not doing anything special.
Yeah, but I guess this will some questions when a commercial product will be developed.
Yeah.
That is a shelf life.
Yeah, I suppose in the future, per sure, we will use surfactants or things like that to maintain in better condition, in natural condition as well. But it's the thing at the moment, we don't know.
There are also questions about what do RNA becomes within the plant? Are they remobilized in the plant? Do they have any systemic activity?
Sorry, repeat me the question.
So are RNAs remobilized within the plant? And do they have systemic activity?
At the moment, we don't know with our target if are able to be transported between the plant. But in other studies, they were proof that some of the dsRNA targets, they can move. So I will say yes. But in our case, we would like to do that experiment.
There are also questions about the specificity of these RNAs. So you tested them on botrytis, but can you be sure that they can have also that they could have activity on other fungi? Have you tested that?
No, but it's in process.
Yeah,
because there are, for example, publication about some, I thought was the beta-tubulin gene of Fusarium that was working as well, the same dsRNA was working in botrytis. So maybe in close, very close related fungus can work.
Okay. So maybe a more general question. Do you think oligonucleotide application is not considered as GM crops? Sometimes it is very skeptical that this might causing mutations to the plant.
I don't know. I think it's going to be part, I hope so, for the different strategy. But we will see, because at the moment, we don't have legislation related with that as well, so we don't know.
Okay. So maybe that would be a big topic. Again,
Yeah,
when some
fair
of the-
true.
Yeah. So the results are for the moment only in the lab. Do you think these results might be replicable in the field? Do you expect much higher RNA degradation in the field rather than in the lab?
At the moment, we, theoretically, yes. But if you put in the nanocarriers, we hope, at least we did an experiment, preliminary experiment in field condition, and we obtained at the moment the same results that we saw in the lab. But you never know.
Yeah, okay. There are many more, but maybe just one last, if you have time. Can you give details about the nanoparticles that were used?
More details, sorry?
More details about the nanoparticles that were used in the protocol.
The bioclade, you can find protocols in the publication of Ninameter, so it's very easy to make. And the carbon dots as well, there are publications, so both researchers, we are in collaboration with them, but it's published material.
Okay. So sorry we don't have time for all the questions. But
Anyway,
I'm
I
sure
will be in the coffee and during the meeting, so no problem.
And please, you can also answer in
Yeah.
the chat. Yeah. Thank you.
Thank you so
again.
much. Thank you.
Thank you.
Okay, thank you very much. Coming to the last, but of course, like ever, never least, presentation from Caspar LANGENBACH from the Aachen University Plant Physiology Department, enlightening us about the identification and utilization of antifungal and defense-stimulating molecules in Asian soya rust. The floor is yours.
Works on its own. Thanks, Helge, for the nice introduction. It's a pleasure to give this seminar here at ICPP in this impressive room. And I hope you can hear me well. And I'd like to report on the activities of the past years in our group and in the recently founded startup that we are trying to really bring to the market to try to manage Asian soybean rust. So many of you will know that Asian soybean rust is quite of economic importance. It's considered to be one of the most important threats to food security and especially causing high economic losses in South America. And this is mainly due because there are no varieties available, soybean varieties, that are really resistant against all isolates of this fungus. And so this requires repetitive fungicide treatments to really manage disease in the field. So we wondered if we could learn from non-host plant species how they effectively fend off this pathogen. And to make quite a long story short, amongst other mechanisms that we identified, we found that non-host species accumulate coumarins upon infection with Phytophthora pecoriaceae, which is absent in the soybean host. And if you have a closer look, you can see that here in these two non-host species that we had a closer look at, Arabidopsis and sunflower, you can see that there is also a qualitative difference in non-host resistance. So in Arabidopsis, the interaction is terminated with the penetration of the epidermal cell in wild-type plants, whereas in sunflower, the interaction is stopped already at an earlier stage during germination and aprosoria formation. And this coincides with the capability of sunflower to secrete coumarins to the leaf surface, which is not the case in Arabidopsis. So we thought, would these coumarins be applicable for controlling Asian soybean rust? And we had a closer look at the major coumarin that accumulates in both non-host species, which is scopoletin. And you can see that scopoletin in vitro assays in a dose-dependent manner inhibits spore germination. And this is linked to its strong antioxidant activity and its capacity to cross fungal membranes. And you can see that in the presence of scopoletin, fungal or reactive oxygen species accumulating in developing hyphae are repressed, so the ROS is repressed, ROS production is repressed, and this is thought to interfere with fungal development. And you can also see on the other, on the right-hand side, that scopoletin, if applied to soybean, really can provide some degree of protection from Asian soybean rust. So it may qualify as a fungicide, as a natural fungicide for controlling Asian soybean rust. But we wondered if we could also generate transgenic plants that would accumulate this compound, and we did this by stably overexpressing the rate-limiting enzyme in coumarin biosynthesis, which is the F6H1. We took the Arabidopsis F6H1 that, when overexpressed, yields to a strong accumulation of scopoletin, so this antioxidant bioactive version. But when high levels are reached, the cell tries to maintain redox balance by glycosylation and storing the inactive form scopoline in the vacuole. And you reach much higher levels of scopoline than scopoletin in Arabidopsis as well as in transgenic soybean. But still, antioxidant capacity of these transgenic lines is severely increased, and you can also see in the lower panel that if you apply mycotoxins like fusaric acid to soybean, you see a decreased accumulation of reactive oxygen species in these transgenic lines. And this is also true if you do the same in transgenic BY2 cells that we generated. So if you apply fusaric acid or DON to BY2 wild-type cells, you get a strong induction of reactive oxygen species, and this response is entirely suppressed in the transgenic cells. And also correlating with this increased capacity to cope with mycotoxin-induced oxidative stress, cell death is clearly reduced in the transgenic lines compared to the wild type. So as a next step, we wondered if this increased tolerance to oxidative stress would also go along with increased resistance to a mycotoxin-producing fungus that is of relevance for soybean production. So we had a closer look at Fusarium virguliforme causing sudden death syndrome. And you can see actually that, in fact, resistance was not altered in the transgenic lines compared to the wild type, but we saw an increased tolerance of two independent transgenic lines compared to the wild type, which is likely due to the capacity of these plants to better cope with mycotoxin-induced oxidative stress. We also found that these transgenic lines, the soybean lines, were less attractive apparently to beet armyworm, so a generalist insect pest. And we also found that high levels of scopoline in roots of Arabidopsis thaliana also rendered the plant less attractive to female nematodes. But whatsoever, unfortunately, we did not reach a level of soybean rust control that we were looking for, so we saw some 5% reduction in disease severity transgenic lines compared to wild-type lines in field assays. So we thought we should better refine our strategy and really try to copy what is going on in the sunflower non-host, so really secrete blends of coumarins, so mixtures to the leaf surface to there suppress already germination of fungal spores. And so what coumarins to choose? So it's important, there are many different coumarins, structurally quite similar, but they have very different properties. And if you want to secrete them to the leaf surface, we thought it would be best to choose coumarins that are also light stable, not degraded under when exposed to light. And you can see here that, in fact, scopoletin and closely related scoparone are really prone to light degradation, whereas, and this is, I think, quite astonishing, isoscopoletin, just the isoform of scopoletin, is entirely resistant to light. And it's also, as said, normally the plant, when high concentrations of these antioxidant coumarins like scopoletin or isoscopoletin are reached, the plant kind of detoxifies this by glycosylation. So the bioactive form just can be produced to a certain limit. And this is different, for example, if you here have a look at scoparone levels that can be achieved. Here, these are transient assays where we overexpressed O-methyltransferases from sunflower and from F6H from sweet potato. And you can see that the levels of scoparone are much higher than the ones of isoscopoletin and scopoletin. So a mixture of these coumarins, we think, is the right approach to really go with. And the next step, of course, was to find a transporter that would be suitable to facilitate transport of coumarins to the leaf surface as it is in the sunflower non-host. And so we had a look at ABC transporters that would be co-expressed in sunflowers that we elicited and that then secrete coumarins to the leaf surface. We identified an ABC transporter that we then cloned and transiently co-overexpressed in Nicotiana bentamiana, and you can see that this increases the concentration of scopoletin in leaf water washes of Nicotiana bentamiana, and this coincides with a higher activity of these leaf water washes to inhibit germination of the fungus. So we think we're here on the right track, identified the right genes, and we're now transferring this into soybean, generating stable lines. I'm very curious to see how they perform, but to tease you already a little bit, we already identified in previous test a stable transgenic soybean line that co-expresses 4 different coumarin biosynthesis genes and really gives us a level of disease resistance that we hoped for, so about 25 to 75 reduction in susceptibility to soybean rust. And so we are confident that this strategy will come at some point to a happy end and may contribute to soybean rust control. But apart from the coumarins, on the way we found that actually in the sunflower leaf water washes, there must be additional components that contribute to the spore-inhibiting activity, because when we digested these leaf water washes with proteinase K, we found that we could restore germination at least to some extent. And this indicates that there must be proteins present and secreted to the sunflower leaf surface that combat also fungal germination. And we did a proteome analysis of different sunflower cultivars to analyze the leaf water washes. Currently, we're producing recombinant protein and functionally characterizing its activity to evaluate, on the one hand, the potential of the sunflower phyloplane peptides for engineering resistant soybean varieties, and on the other hand, possibly using these for foliar protection, or use as a foliar fungicide to protect soybean. But as they are very easy to wash off, I mean, the assay was based on this, we think that it also might be a good idea to then, if we identify very active phyloplane peptides, to fuse them with what we call anchoring peptides that we characterized some years ago. So these are peptides that can immobilize antimicrobial peptides on the leaf surface to better withstand wash-off by rain. So, but last but not least, I think, the best way of controlling basically any kind of disease is by combining different modes of action to really achieve sustained protection. And this is why we think it's also very important to have a look at activators of disease resistance, so ZAR activators, or what we now call activators of crop resilience. And to support this development, we recently founded a startup company. We also have a booth here. Meet us, please, at booth 28. And we are committed to identifying and characterizing any kind of compounds that really stimulate plant defense. And so we think that we also have two pieces of what we do in the company. On the one hand, we provide services for partners in the ag industry, helping them to identify or better characterize compounds with such qualities. But on the other hand, we also use our own pipelines there to identify and characterize our own molecules and hopefully bring them to the market. And two examples I'd like to show you shortly that we think also have the potential to contribute to Asian soybean rust control. The first one is a near-natural molecule that really provides broad-spectrum disease tolerance. So it does not only provide enhanced resistance to Asian soybean rust, but it also provides reduced susceptibility of sugar beet to Cercospora beticola, also active against powdery mildew in wheat, and it also provides protection from bacterial and viral diseases, and also showed to promote drought stress tolerance here of sugar beet. The second molecule that we think has a high potential is a natural microbe-derived molecule that was not originally identified by us, but by a partner. And it is, on the one hand, directly antifungal. And you can see that it very well controls soybean rust disease. So the positive control here is a synthetic fungicide, and the level of control is about the same. And we found that it's not just directly antifungal, but it also primes cells for enhanced accumulation of phytolexins. And it can also provide resistance to bacterial pathogens, although not being directly bactericidal. So suggesting that we have here a molecule with dual mode of action, and hopefully this could be another weapon to fight Asian soybean rust in the future. And with this, I'm over time. I'd like to thank everyone involved in this project for funding this, and you for your attention, and I'm happy to take your questions. Thanks.
Thank you, Caspar, for this presentation. Very interesting. Are there questions from the audience here in the room? There are some in the chat, as usual. Why another non-host Arabidopsis cannot secrete coumarins?
Why it cannot secrete coumarins?
Yeah.
So the first, why can it not see? I mean, it's of course a complicated question. We see that it does not. We also see that Arabidopsis does not trigger, or the coumarin production is not triggered during the initial penetration, but the fungus needs to grow into the mesophyll tissue, and there coumarin production is triggered. And I think this may already explain why we don't find it on the leaf surface, but it might also be that Arabidopsis is lacking the right transporters. They might not be expressed, and I mean, it's still a matter of debate how these secondary metabolites in each and every case end up on the leaf surface. It might also require, for example, specialized trichomes that are necessary for the secretion, and these might simply not be present in Arabidopsis.
Have you ever thought on what kind of toxicity comes to human and other beneficials comes from these compounds?
Yeah, so we have not tested, but I mean, we collaborated here with a large partner from ag industry, so with BASF, and they, of course, did some tox checks. And actually, what is reported in the literature, you can see many reports, like on many secondary metabolites, there are widely positive effects associated with on human health, like anti-diabetic effects and so on. So actually, it would rather qualify maybe as a nutraceutical benefit to produce more scopolamine in plants. So I would say.
How far did you go into the field testing already, or?
Yes, with the one transgenic line, with the overexpressor, we went to the field. We did not see the level of protection, as we said, or that we at least aimed for. It was about 5% reduction in soybean rust severity. This is not enough to justify deregulation of this technology. So now we hope with our refined strategy we will achieve this. And I mean, I showed you one very nice result, and I think we will then move, of course, at some point with these lines to the field. But as you know, this requires some time transferring transgenic material, propagating it, and then testing it in a region where you have soybean rust. So we fortunately don't have it here in Europe. So we need to go to South America to test this.
Is there, beside of the perhaps not sufficient activity in the field, any other trade-offs of these GM lines, like yield?
So
quality?
far, we didn't see any obvious, let's say, trade-offs. So in our lab assays, we never saw any differences on seed set, biomass production, and so on. And real, let's say, yield trials have not been conducted so far. I think once we have identified a transgenic line that really gives good results, then this, of course, will be evaluated. Sure.
Have you ever tested cinnamon essential oils which contain coumarins?
No, so
Not done.
I mean, I don't know about this. So, but yeah, maybe if the person asking the question comes to me, we can discuss, and he or she can tell me more about it.
Relating to an earlier, there's a question about a relation to your earlier work about BRT1. And this could confer durable resistance. Have you changed? Have these results changed anything in your work?
So as I said, I focused now on really the coumarins in this presentation. So we also reported on different genes that we transferred from the non-host to the host that gave some positive results. But as it is, you always have to focus on some topics, and we did not have funding or possibilities to further follow up on the BRT1 story. Unfortunately, I would love to do this.
Good. Thank you very much. And there's no other question, then thank you, and then we are going to close.